first thing I notice is anytime anyone's up for coffee, she goes. We might ask her, we'll see how fast she goes to the talk and see how many cups of coffee she's had. And then the second thing that I notice is she's from Wyoming and spent a lot of time in Colorado, and just like me, she thinks that there's an awful lot of hype and panic over the snowstorm that's supposed to come today, and we've been waiting. Anyway, so probably some of you know her better than I do, but I'll do the best I can. So she comes to us from the University of California in Santa Barbara, where she's been there since 2002. And my sense is she started her research really looking at oceanographic features and temperature and how that relates to the distribution of organisms. And so hence she was looking at a lot of oceanic conditions, and she can tell us exactly, but it seems like in about 2005, she started getting really in the early days of ocean acidification, but coming at it from a very different perspective, and that is coming at it from really a biologist and a physiologist. And so I don't know if this is true or not, but I have this vision back in 2005. She's sitting in a room, and there's all these oceanographers, and she's a biologist. So we'll see today what she has to say. I think just by the title, we're going to find out what the individual species can migrate at the lake. That's right.
Thanks very much. Well, so thanks for coming out today, because I know it's a little cold and blustery out there today. And I want to say thanks to the organizers, because this is one of the most nicely organized series that I've ever spoken in. And frankly, it's genius to have the speakers stay in one place and have everyone come to them, so that's really great. And I am wildly over-caffeinated. I will confess to you that not only are you wonderful hosts, but you people drink a lot of coffee. So if I'm tremoring a little bit, it's not because I'm nervous, but it's because there's something pharmacologically wrong with me. So Lisa's right. So I'm an ecophysiologist by trade. I study how organisms function in their environment, and we're interested in simple things like thermal tolerances, all the way up to transcriptomic analyses to look at variable gene expression and what that can tell us about metabolic response. And in 2005, I was invited to come talk to a group of oceanographers who were having a workshop at Scripps. And I felt like the biology lady or something, and it was really wonderful because at that point I was introduced to a fantastic group of people, one of whom was Richard Feely, her very own. And since then, I became sort of incorporated into trying to do experimental work to look at what the effects of an acidifying ocean might be on marine calcifying organisms. And for me, that would be benthic marine invertebrates. So I was working on sea urchins and mussels and some things we like to eat, like oysters and abalone. So I started doing this work. And what I wanted to do for you today was give for you a presentation that is sort of how I'm starting to think about this field. And so the whole thing really starts right here. Our problem really is about atmospheric CO2 and an anthropogenic release. We like to burn things, fossil fuels in particular, and so recently at Mauna Loa Observatory, they recorded 390 as sort of a median level there. And I can tell you when I started working in this, some of my first slides where we had control levels of CO2, it said 384. And so in the time that I started working on this, which I find personally alarming, I've been changing my slides to show the increase in what we think is average atmospheric PCO2. Now, I'm not going to go over too much Ocean Acidification 101 because you've had so many luminaries in front of me. But what we do know very clearly is that increased atmospheric CO2 translates into dissolution into surface oceans and a change in oceanic pH. So that's where we kind of leave our heroes on the oceanographic side and how we think about oceanic atmospheric interface. And my brand of science was then to ask this kind of question. Given that organisms in their environment have sort of some biological abilities to resist, tolerate, or compensate, a particular phenotype is able to react to changing environment somewhere along this scale, and you can adjust this time frame. It's either in a simple laboratory experiment on Tuesday, or it's over an evolutionary time frame. at some point, we want to know, is there a tipping point? Is there some PCO2 or microatmospheres of carbon dioxide in seawater at which function X fails? So reproduction becomes a problem. Most of the time we focus on calcification process. So where is this tipping point? And these kinds of studies have ecological and economic importance, and so this is the reason that people are going after this. And so the way we think about this as a biological problem is that, and this is my homage to ocean chemistry, Andrew Dixon here, if you could just be amused because this isn't very detailed, but here's how it comes out in the wash for me. CO2 dissolving in the seawater shifts carbonate chemistry such that there is a lower pH, which physiologically organisms then have to deal with. It might alter their own intracellular pH, it might alter dissolution processes of actual shells exposed to seawater, and it lowers the carbonate ion. So this is all reviewed for you guys because you've been in the series, etc. And this is really where I came into the field in 2005 because we were interested in these processes. And what I want to do for you now is sort of do the brave and or stupid thing, which is to not, I don't want to stand up here and show you slide after slide after slide of what happens to calcifiers, but I really just want to have a conversation with you and tell you what we've been thinking about lately, because there's been a lot of motion in this area in global change biology in the oceans to try to think about how we can approach these things in a sound experimental way with really good balance, and to also recruit people from other fields. So how can experimental evolution help us answer these questions? And isn't there an incredible role for population genetics in these issues? Because a lot of the stuff you're seeing right now in the literature, admittedly from my own group, is sometimes it's not a nice name, but it's like impactology. We put species in here, we see what they do, and we're really just testing variable pH or variable PCO2s on extant population known genotypes that are here right now, and we don't really know how these things will play out in the future. So these are the kind of thoughts that I've been having, and rather than do sort of a presentation on organisms, I wanted to focus this in a different way, a way that is a little more nuanced and a way that will allow us to talk about broader issues than just that calcification is threatened. Because my real message here is that this is a big issue. Ocean change is an issue, and we can use the input from many, many fields, all the way from comparative genomics to energy, management sciences and fisheries, and in the middle, population genetics, etc. So the more the merrier. That's kind of my message. So, but while I go through this, I'm going to try to show you examples that do touch upon some data that we do have to illustrate a couple of points. And so this is the big map of how I want to talk about this today. Today, I'd like to wear my global change biologist hat. And global change biology is an intersection of the environment, the organism, how it interfaces with the environment, and then what will happen to the ecosystem as the environment rapidly changes. And there's a journal, Global Change Biology, that kind of publishes these types of studies in all biomes, right? So if you ask a global change biologist how organisms respond to environmental change, whether they work on plants or birds, these will be kind of the answers that you get. There's migration, acclimation, or the proper term is probably acclimatization, adaptation, or extinction, right? So this is really the mold into which we're really starting to think more about ocean acidification impacts on organisms. And I'd like to take us through how to think about this because some of the answers are very important for various ecosystems, and they're not the same for all ecosystems. So let's first consider migration, right? So the obvious answer is if you can't take the heat, get out of the kitchen, go to a different environment, find a place where you can reproduce, where there's available food. And there are some pretty serious consequences for this. These responses might be limited by life history traits, available food, available habitat, or you need a predator that's just not going to support your survival in a certain area. It'll certainly drive species range shifts, but one of the things that this highlights for us is that this really, in many cases, might not be at all possible in ocean acidification scenarios. because you run into the double jeopardy problem, organismal responses to temperature and acidification together. And my little case study here is our pteropods, sea butterflies is their common name. So this little guy is, many of you probably know this because of what you do and what school you're in, but it's a pelagic mollusk. This is Limacina, helicina, Antarctica. This is the form that's found in the southern ocean, but there's a very closely related, probably a subspecies, people think, that's Limacina, helicina. And they're an insanely abundant part of the zooplankton, and as you all know, they're really important in short food chains. In the Antarctic, that's absolutely the case. These things are primary fish food, and the fish are food for just about everything else. That's there, seals, whales, etc., and other bigger cruciferous fish. And so one of the things about the pteropod is that it's a shelled individual, and here you can see its little intestinal material, and it's harder right here. And there's this beautiful crystalline shell and a modified foot, like a regular snail has a muscular foot that it moves along on. And that's been modified in this species into wings, into a little sea butterfly. I don't have the video with me today, but it kind of flies through the air. And they're about the size of a peppercorn, and they appear in clouds. And the first time I saw a pteropod, I have to confess, I was working on there on fish, and one of the fish barked after we caught it, and it just brought up this mass of little crunchy, tiny shell things. And I thought, what is this? And it turns out that the gut contents was like 80% pteropod. And so one of the fears is that these guys are right on the hairy edge of being able to perform calcification. So they're kind of difficult to work on. Either you get them from shipboard in the Arctic, but in the Antarctic, we drill holes in the sea ice using a gigantic reed drill. You can see the bit right here punching through the sea ice. And this is me on shovel over here. And it's not very glamorous. I just use you don't really need, they're so abundant, you just plankton tow for them. But you always have company, so there are Liddell seals hanging out watching you. And you can see this blood right down here. They get in and out of the hole and they get cut a lot. And there are these Antarctic conservation laws that sometimes you actually have to stop work because they show up in your hole. So all we do is plankton tow for them. And here's another work stoppage event. But once we get them back to the laboratory, here you can see them in a little jar. At this point, I was collecting material to do some 454 sequencing on them, which we've been doing. And we plan to use this to look at whether or not they can respond effectively to temperature and acidification. So we're making molecular tools for that right now. But they're just beautiful and teeny tiny. And I tried to make an art film of them swimming in music, but I didn't have the right equipment. But they're incredibly beautiful, translucent shells. And we don't have very much calcification data on them right now, but one study that we do have that we know about in the literature comes from McMurdo Sound, and they were working on the Arctic species. This just came out recently. And if you hold the pteropods at lower pH of 7.8, which would mimic a high CO2 environment, their calcification levels drop significantly. And this is a pretty big change from standard incorporation of calcium carbonate, and it drops off significantly. And this only occurs over a couple of hours. So they just put live pteropods into a jar to see what would happen. And so this is a great example of sort of an impact experiment where we see that the current genotype-phenotype combo responds negatively to this really acutely applied stress. And over time, we think these stresses might occur over a much longer time period than 7 hours. But relative to geologic time, the change is coming incredibly quickly. So there's a lot of concern that pteropods will not do well. So if we go back to our migration phenomena, this is just a map of the southern continent of the Antarctic. And the water around the continent is minus 1.9 degrees Celsius. There's no temperature climb. It's just cold as can be from top to bottom. And these things are very cold adapted likely. So if these organisms were to swim out of colder waters, they would be moving into, they'd be going north into warmer water. And we're not really sure if polar species have sort of the adaptation poise to maybe escape acidification that is occurring in the Southern Ocean and head for New Zealand or Tahiti, to find better carbonated water. There's probably a limit to where they can go. So these kinds of phenomena so for this community, it's kind of interesting because these are really, really difficult issues. There are climate envelope models that look at the distribution of the species now and say, if temperature changes by 2 degrees, the species will move this far, but it doesn't really incorporate the natural tolerances of the organism. Can a pteropod actually move to a place that is 0 degrees, plus 1 degree, plus 2 degrees? And so this multi-jeopardy phenomena in climate change biology is it's an area that people are beginning to really think about. There's more need for multi-stress research, and I talked to some people here that are working on it. But this is to highlight a serious gap in our knowledge, is how do different stressors come together in the environment, and migration possibility highlights that. So this one kind of highlights probably the research that I do the most, and that is acclimation or climatization. Do organisms right now, do current genotypes have the variability in their physiological responses and potential phenotypes from those genotypes to tolerate what are pretty rapidly changing environments? So whereas people might criticize impactology, like, why are you working on urchins now? Because they're not going to be the urchins that are around in 2100. But because the environment is changing so quickly, it may be that a lot of metazoans in particular don't have time for adaptational change. So in many ways, exploring the tolerances of populations now with tools of population genetics and physiology, population biology, is a really, really good idea. This is something that we should be doing. And as a functional biologist, what we're asking is, are there conditions under which species just kind of pop out of their tolerance windows? And this is something that my postdoc and I, Ann, Todd and regroup for review. But this is like classic biology from 100 years ago. You can go into the literature and find the idea that here's a cold thing, we acclimatize, you're acclimated, and its performance shifts like this. And then these are the windows I'm talking about, that different processes have different tolerances. So on the inside, growth and reproduction might be the most sensitive. And then next comes their activity, and then there's, maybe I can just barely tolerate this. And then resistance abilities are the most furthest out from the norm. So these are classic ideas that are now being explored in a climate context. And so what I want to do for you now is just kind of show you a little bit of the data that's coming out of the lab. So one of the things I wanted to do when I started working on these things was to choose a species that would give me some genomic tools as well, but it was a benthic marine calcifying thing. So we've been working on sea urchins. We spawn them in the lab, and then we raise them under high CO2 conditions. And here they, develop, in this particular species of purple urchin, and it'll develop into a larvae in the lab in like 3 to 5 days, depending on temperature. And as I'll show you later, they're calcifiers. These little sticks you see in here, they're like a backpack tin. Those are calcium carbonate rods that a freestanding backpack that then has all this tissue around them. And so they're the perfect little system to work on because they're calcifying, but we can raise a lot of them. We can do crosses, make half sieves, etc. And so the way we do this in the laboratory is kind of interesting. Like, I didn't want to build a facility, and I wanted to go to the Antarctic, and I wanted to go to tropical places. So we built a system that was portable. And I wanted to show it to you here because it's the most MacGyver thing you've ever seen on the planet. And this is set up in the Antarctic. But it's cool because all we need at the other end are buckets and a tank of CO2. So there's a tank of CO2 in the lab, and tubes are coming out of it, over to a place where we scrub moisture and CO2 out of the air using those columns of fire soda absorb. The gas is then mixed with these commercially available mass control valves, And those different colors represent control, middle, and high levels of CO2. And we run them up and out over the ceiling and right out the door of our laboratory. Carefully drilling a hole in the wall that we didn't caught doing. And then we use that gas to create reservoirs of water that have a set microatmosphere of CO2. And eventually we put zillions of buckets. I shot this pretty early in the project. We put tons and tons of pickle buckets that we then use to raise our larvae. And so the reason I show this is because this was supported by NSF, and it was built by a bunch of people working together. It only cost us about $15,000. We can put it in a suitcase and take it wherever we want to go with it. It looks terrible sometimes when we set it up. This is the setup in the lab in Santa Barbara. But this shows what happens. We have a set known level of CO2 in our water, and we can do appropriate biological replication and technical replication, and these are buckets of larvae with motors on them. And so we raise the larvae, we fertilize them, and we raise them from a single cell embryo all the way up to something that has a skeleton. And so, as I said, larvae have that internal skeleton. They have a kind of calcium carbonate that's called calcite, so it's kind of dissolvable. And we know a lot about how it's made mechanistically. The interesting thing about them is that even after about 18 hours, the gastrula are starting early calcification events. So you can see the beginning of this teeny tiny skeleton has a little jack structure, and then it grows into these bigger arms, and the Polius larvae just expands. So in experimentia, we can follow calcification of thousands of these little guys as they develop under different CO2. And so the species that we're working on a lot right now is the purple urchin, and its cousin, Strongylocentrotus franciscanus, is the fishery species that California divers go after to provide the roe for the sushi uni. And so we collaborate with the sea urchin divers in Santa Barbara to kind of think about what acidification in our neighborhood will do to their recruitment processes, and it's turned into a kind of a cool collaboration. So one of the things that I want to show you now is that before the moment that I'm standing in front of you, like maybe July, we were doing our experiments by creating seawater that was set at what were IPCC predictions. So there would be one prediction, the B1, that would say, well, by 2100, the atmosphere will go from 380 to 570, or a really high business as usual scenario like this to 1,000. And we started to realize, I mean, as a physiologist, I was realizing that, we should be able to know more about the environment, and I wanted to know more about the environment, but there was very little available technology for the average person like me to get that data if you didn't have an oceanographic buoy and these devices called SAMIs and a lot of expertise. There's almost no data, and there's no equivalent for the hobo temperature logger, the tidbits, iButtons, things that we can all use to get temperature. And it became really clear that the upwelling process on the coast was changing the water chemistry on a seasonal basis during the upwelling season from April to October, where the purple urchin lives on the entire west coast from Banfield to Baja. And some of these data actually were brought to my attention by Richard Feely, who instantly called me and said, hey, you ought to see our data in our science paper that shows these big fluctuations on the coast where upwelling water is shoaling. Some colleagues in Oregon put out some sensors off of the central Oregon coast and gave this to me in about 2009. And here's April in time going over to October, so there's the spring upwelling. And these numbers, the red is 1,200 parts per million CO2. So remember, 1,000 is pretty high for IPCC predictions. This is the surface. Here's depth down to 100 meters. So we think our larvae are always right around in here. And this green is about 800 parts per million CO2, which is fairly one of the dire IPCC predictions. So instantly we realized, this upwelling process on the coast is creating a lot more natural pH variability than we knew about because we just weren't paying attention, we couldn't detect it. And so, I'll just skip over that. This is my little upwelling cartoon. But when you go to the ocean, pushes deep water up that has a lot of stored CO2 in it, and that low pH, high CO2 water shoals up onto the coast. In many cases, it comes right to the surface and right onto shore, so that it's greeting a lot of calcifiers like urchins and oysters that might be in hatcheries. And potentially one of the problems, I know that Dr. Feely described this to you, some of the oyster issues may be the acidic water coming into hatcheries, and this is how it's happening. So this is like, you know, sort of a very, very, very new thing for the entire research community. So I really wanted to be able to get these data in my own backyard so that I could ask questions about what pH was in various places that I was working. So a colleague of mine named Todd Martz, who's at Scripps, is kind of an ocean engineer, and he had worked at MRI with Ken Johnson and some people there, and they built sensors. Basically, it's the coolest thing in the world. It's a plastic tube that has two Honeywell Durafet pH electrodes poking out of it. And these copper cages are protecting the glass points of the electrodes coming out, so there are two reference electrodes, so you know how to check for what they measure. And it has batteries inside of it. It's sealed with screws so no water gets in. It can store up to 11,000 data points, and you can program it to come on and take data from whenever you want. And the batteries lasted for, we don't know how long, so we were beta testing these for Todd, and we deployed it off the coast of Santa Barbara at one of our LTR sites. And again, came up with some rather eye-opening data that makes me want to say two things. This does not mean that ocean acidification is not a problem, but it means that we need to consider the environmental background and the evolutionary history of the organisms we study, because not all oceans are created equal. We know this for sure. And each calcifier is experiencing a different kind of environment out in the world. And that alone might influence whether or not it will be able to adapt to future changes, or whether there are genotypes in the population that will respond really well. So this is where population genetics theory can be handy. So these data are just coming out right now in GMD, but here's the pH profile off our coast. So, you know, we have variation from 8.2 down to 7.7, and the PCO2 for this run of only 3 weeks in the summer went from 250 microatmospheres all the way up to 750 microatmospheres over a 3-week period. This is kind of a kelp-forest environment, so there's a lot of activity in the water, but it's just a real message to the community now. This is a really important thing for us to do. I don't want to call it collect observations and do monitoring, but to be much more informed about what experiments we should be doing. So we're starting to think that we don't want to parameterize our lab experiments with IPCC projections, but we might want to do it now with values that we're measuring out in the field. So these are some data from the lab where we took purple urchins and we raised them in that system. And we measured parts of their growing skeleton, which is a great proxy for growth and development. And you just kind of add up the length of, in this case, the left side of the total skeleton. And we raised these guys at 3 different PCO2 levels. And they kind of bracket the levels that you see in the water. This high is the highest that we reported in Oregon. And what you see is that there's a decline in growth. So probably metabolically, we know they're struggling to calcify. There's a cost of developing in this caustic water. But note that they're developing just fine. They developed stomachs. They weren't little hopeful monsters. And so, you know, there's some suggestion that the way we should be thinking about this is that in their natural environment in an upwelling coast, they might get trapped in an upwelling event for 5 to 7 days, let's say. And there's a cost of having that happen. Probably a lot of ion pumping, some deleterious impacts on making their skeletons and developing, and they end up in smaller individuals. But they're not killed, this particular species. Now the thing that we don't know is what happens to these littler guys when it comes to settlement time. Are they as successful at settling? Do they become smaller adults? The other thing I can tell you is that there's a genetic component. I'm not showing the whole array, but if we raise subfamilies, different mothers, different genotypes have very different responses. So that there are some genotypes out there, some females that are creating progeny that are better at tolerating these conditions than others. And so looking at that component of response to ocean acidification is really important. Now, the next really emerging area in the study of organisms and ocean acidification is to really try to understand not just the phenomenology, like, oh wow, you're not calcifying as well as you're smaller, but what are the costs? And so we're sort of beginning to approach this in the field, and in everybody's lab, people are looking at this. But these are some really early data that Michael O'Donnell collected. He's now up at your Friday Harbor as a postdoc and fellow researcher. And it's just to describe it, Moose raised red urchins under different CO2 conditions. And he then took the larvae and he heat stressed them, asking if you grow up as a compromised individual under these difficult low pH conditions, does that alter the larvae's ability to tolerate future stress? And so we really stressed them in kind of an unfair and not ecologically way. But we heat stressed them really dramatically. And what we found, the blue's control and the orange or the high CO2 raised larvae, they were really unable to upregulate one of their most important defense genes, a molecular chaperone called HSP7. And so this suggested that they might be compromised functionally in terms of how they would respond or survive temperature stress in the field. One of my other postdocs, we're still kind of exploring the depths of this, but one of the other postdocs then took larvae, raised them, and gave them a thermal challenge. So on this axis, here's the control guy for 380, so they grew up for 5 days under 380 conditions. And they were heat stressed for an hour, and about 15% of them died when you get to this really high temperature. And then it compared them to larvae that were raised under high CO2 conditions. Many, many more of them died. And we think it's because in some way their gene regulatory network or their ability to activate these defense genes are kind of compromised, or there's something going on. And so this sort of continues to highlight our physiological plasticity work. We then went one more step and used a microarray. And I promise this will be the last weirdo molecular biology thing we talk about. But we wanted to know, if you have a genome and you're turning it off, the genes off and on in a particular way, if we look at all the genes for different processes, can we see signatures of stress? Can we see that calcification genes are going up as a defense mechanism, or molecular chaperone genes are going up as a defense mechanism? Because you can see what sort of things you're doing. So we created a microarray that had a lot of genes in all these different categories, so biomineralization, acid-base regulation, and stress. And then we just screened gene expression, levels of RNA, using that chip. And I'll just show you this one piece of data, but what we found in the purple urchins was at high CO2, they weren't freaking out metabolically. They weren't going, oh my God, we are going to die, as they would in response to temperature stress. They actually did metabolic depression so that they downregulated some of their genes that were responsible, in this case, for the skeleton formation. These are all proteins, transcriptional proteins that are part of the skeleton. And these are some regulatory genes over here. But they were kind of turning down the pilot light. And this is me totally making up stories here. But we had this data before we knew about the environment, and we kept saying, wow, it's almost like they're hunkering down and not being as metabolically active under these high CO2 conditions. And so it makes us think that the larvae are somewhat adapted to do this. A lot of marine invertebrates have metabolic depression in their toolkit. They have diapause stages. So one of the things we wonder is that when these larvae are out in the water column, while they're out there feeding for 4 weeks, let's say, if they get trapped in one of these masses of corrosive water during an upwelling event or something along those lines, they do metabolic depression in order to not keep metabolically struggling energetically against these harsh conditions. So this isn't that far-fetched, because you can go in the literature and find quite a few examples of this. So here we've got a case where I've sort of got like a new light shown upon the experiments that I did on purple urchins, because we now know more about their environment. So I want to show you some data from a different location, in this case, the Southern Ocean in the Antarctic, to go back there. Because as we are able to understand more about the current environment, the pH exposure of organisms and what they might experience in the future, you start to realize that all oceans are probably not created equal. So I want to show you some data about urchins from the Antarctic. And these urchins I want to show you are coastal organisms. They're like essentially in the identically same benthic depth habitat that the purples are on this temperate coast of Washington, Oregon, and California. The species is called Sterechinus neumayeri. And again, you know, we flew down there with the U.S. Antarctic program. We drilled holes in the ice and we put fish huts over the holes, like ice fishermen in Wisconsin kind of thing. And then we used track vehicles on ski slopes and piston gullies to drive around out there. So we actually locomote around on a frozen ocean. Kind of strange. But this is Sterechinus neumayeri, and this is a spawning event. One of our divers caught several spawning events in nature. So while we were there working in October, this is their peak spawning time. And there are really abundant organisms on the bottom, and they're very similar to the purple urchins. They're an obligate feeder in the plankton. And in the Antarctic, cold polar waters are really cold. Physically, they hold more gas. The thought is then that their PCO2 is much lower, their pH is lower, and as more anthropogenic CO2 dissolves into the water, people think Antarctic ecosystems are more vulnerable. This has been one of the predictions we had. But really, we had precious little data about what the pH actually was in the water. So when we went down there to raise urchins, we didn't really know what control log meant. Like, what should we set it at? 350, 380, 420? We had no idea. So we were able to use the CFET to do that. And we were working, here's McMurdo Station. We had 3 sites that we recorded pH data in, 2 coastal and 1 more out in open water. And the depth in the open water is like 500 meters. And this is what we get. I haven't had a chance to really plot this up really well yet because we just recently got back. But this is 7.9, and there's 8.04. And the red and the blue traces are the 2 electrodes in the CFET, but there's some noise in the signal, but it's reasonably unchanged. And if we plotted it, this is only a few days, if we plotted the entire time, it's just relatively flatline compared to the coastal site. So there's precious little variation in the pH in the Antarctic site, and the pH is, you know, a little bit on the low side, around 8.04. When we raised our larvae, and these data we're just analyzing right now, we've noticed that they are a little bit smaller. This is the 800 level, 550, and this is control that we set at around 410, because that kind of matched the more acidic pH. And we think that there's about maybe a 10 to 50% impact on growth. But even though there's not a lot of variation in that pH, these urchins are showing us a lot more resilience than we thought. And so if you plot the data of a 30-day fluvii, there's a little bit of a difference, but it's not really significant. So I asked myself, why on earth? Because I guess I was just stereotyping them, and I thought, God, certainly they should really be catastrophically affected by this. And then I turned to my oceanographic colleagues who had worked on some data for cruises and JGOFs, and it looks like what they're finding further out, not under the sea ice where we were, but further out, that there are huge seasonal fluctuations. So that we were working there at a time when the pH was quite low, but that it starts to alkalinize as the season goes on. There's some deep mixing that comes up, and there's actual pH data that I should have for you right now, but I don't, that shows that there's a lot more seasonal variation in the Southern Ocean than I knew about. So what we need to do is keep our loggers out there for a winter cycle to see what's going on. But this is a classic example of how I stereotype a stably adapted organism to think that it won't be able to tolerate these conditions, and in fact it did. So knowing your environment is really helpful. One of the last pieces of data here is actually a very important area. And I don't want to ramble on too much about what to do here, but graduate students, undergraduates, people looking for projects, this is a really cool thing to do, is to look at fertilization, because it is the first step. And for many, 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 many organisms, this really does truly go on as a very quick event in the water that the broadcast spawning or the spawning occurs in. And we need more data like this on more species. And so this is a fertilized sea urchin embryo. In the Antarctic, we've been working on our technique to make sure we have good statistical design and enough replication. But I wanted to show you this picture because this is crazy. These are little vials of eggs that we're fertilizing, and this is Colleen New, Lydia Katzenberg, and Mary Sewell, who work on the project, married at the University of Auckland. But we have to keep everything on ice. So we're actually like fertilizing and desperately trying to keep everything cold. And in fact, ice is about 0 degrees. So we're heat stressing our eggs as we do the fertilization because they really should be at minus 1.9. So we've built an incubation block. But this is always a great image for me because when we work on temperate species, we don't do this. The biggest struggle down there is keeping things cold enough in a modern environment and a modern laboratory. So interestingly, though, when we look at the fertilization data, when we have a much higher level of CO2, around 750 to 800, there is a drop in normal development and in fertilization that we see. This is sperm concentration, don't worry about that. But there is an effect once you get to a really, really, really high level. But at 400 and the mid-level, about 550, there's no difference. So I can't make too much about this yet, but it really does suggest that there's a lot more resilience in some of the processes and organisms that we're studying than we might have expected. So one of the reasons I really focused in on the Antarctic to go there was because in 2008 there were some papers that came out predicting that when the atmosphere got to 450 parts per million carbon dioxide, and we're at 390 now, so it's not very far away, that there would be some seriously catastrophic changes in seawater chemistry for polar organisms. And certainly, we're thinking that that could be very, very true for pteropods, which is very, very bad for the food chain. But it's quite possible that for other species, they might have more resilience than we actually think. So, again, this is kind of an argument for needing a lot more organismal work because right now, the single species studies are few, and it certainly doesn't tell us very much at all about interactions, species interactions, or ecosystem level changes. So the last thing I wanted to show you is absolutely, without a doubt, sort of one of the unexplored frontiers in global change biology in oceans. And so much so that geosciences at NSF asked a colleague of mine, Dave Hutchins, at USC, he's an oceanographer, biological oceanographer, and myself, to organize a workshop to think about how can we bring evolution into the study of ocean change. And 2 days ago, we just filed a report with NSF. We had a hoedown with a bunch of marine scientists, biological oceanographers, and evolutionary biologists to kind of talk about how to do this. And so, really, the question is, literally, do we have the techniques right now, the theory in evolutionary biology, and the wherewithal to ask these questions about critical marine species? And it's a question of rapid evolution, because relative to the conditions, the way that the organisms have evolved to this point, the environment, especially with acidification, is going to change incredibly rapidly. And so the question is there theory or methods out there in the world we can use to address these problems? And so I'm going to include this as an example of, well, is adaptation a possibility? And this might be how we would study it. This comes from a colleague of mine, Sinead Collins, who's at the University of Edinburgh, and she's an experimental evolutionary biologist. And Sinead really thinks about rates of change and whether, and she's a theorist, But she does experiments too, so she raises Chlamydomonas in little cultures and asks, okay, if this Chlamydomonas culture is at high CO2 and this one's at low CO2, and they do tons of generations and with the potential to adapt, do we actually see changes in fitness? So these are appropriately designed and well done experimental evolutionary biology experiment that's published in 2010, and she's got a great paper synthesis paper that just came out in Evolutionary Biology, if you want to read about these ideas in greater depth. But basically, she was able to show that the control strain that everybody got about 320 generations had lower fitness than the evolved strain. And these are just relative numbers, but basically, adaptation and evolution didn't happen. So that here's an example of a method, an approach that can be used in marine sciences to look at these things. And I'm not enough of a theorist to really go on about this, but it really argues that there are collaborative efforts, there are ways to look at different genotypes, to use QTL mapping, and to use tools that exist right now to ask really important questions in global change biology. And certainly one of the newest efforts that I'm involved in right now is a collaboration between a bunch of oceanographers at Oregon State, MBARI, and UC Santa Barbara. And this is a project that was just recently funded by the NSF Ocean Acidification call that came for proposals. And what we're trying to do is ask about local adaptation. So instrumenting these different sites up and down the West Coast and looking at the physical environment, what's happening there, how much corrosive water is coming up with what regularity. And then Steve Columbia at Stanford is going to be doing some genotyping work, and we'll be doing some physiology, to just simply ask, do we see performance differences that might have a genetic component where we have local adaptation of benthic marine calcifiers? In this case, we're studying urchins and mussels because we have tools. Just to ask, can local adaptation occur to this component of the environment, pH and altering carbonate ions? So this is looking at what we're calling the ocean acidification mosaic, or the pH seascape. This is kind of where we need to go to answer things in a much more balanced way and in a much more informed way. And certainly, to close, I think that for this community in this room, and for things that we can study right now over the next 5 to 10 years, you will likely be seeing, a more of a push to recruit more people with your kinds of skills. Certainly, everyone that I talked to today has molecular tools to population biology tools that can be applied to these kinds of questions. And for me, you don't want to think about extinction, but it's these two in the middle that really hold a lot of, I want to say, hope, because as we approach this as a society, what we're really asking about is vulnerability. It's vulnerability about things we eat, food security, what species are important to us, and vulnerability of really valuable ecosystems. And understanding more about these potential responses is really key to that success. So just really quickly, this has been a labor of love, a wildly collaborative effort between lots of people in my lab. It was a little foray into seeing what CO2 did to things, and it's turned into quite a project. And we have a lot of fantastic colleagues. Andrew Dixon helps us with our seawater chemistry, and Richard Feely. And the work is largely funded by NSF, but also by a University of California-funded consortium that allows some of us at Davis and Scripps and Santa Barbara to work together on this issue. So thanks for the conversation, and so I'll be happy to answer questions.
In that 320 generation experiment,
Yes.
it seems like at the control there was a lot of variability in responses, but in the experimental one there was very little variability in responses. Does that indicate kind of at the limit of, you've selected all the change you can select for, and there's nothing more to change.
Yeah, I wish I could call Sinead right now and get her on Skype, because that's right. So there are some examples in the literature where you can go and see that things that are already broadly adapted, they have already maxed out their ability to change, or that over many generations converge on one solution. And it's a really important concept, because if it plays out one way or the other for different populations, it's really dire. So let's say that purple urchins or native oysters that live in really variable environments right now don't have a lot of evolutionary grist for the mill. They don't have a lot of evolvability or plasticity left in their genome. So as acidification comes along in these environments, the CO2 that's upwelling has stored carbon dioxide in it. So some of the anthropogenic gas in there is probably 50 years old. So as time goes on, that low level, that trough line on my graph is going to be 7.3 now, pH, but it's just going to keep going down, down, down. And what we're really asking is, can organisms expand that bottom part of their tolerance window anymore? And it could be a really bad answer. It could be a bad outcome, but the answer is no. So one of the things that was really interesting to me was I had a conversation at a recent workshop, and there is some thought that coastal upwelling regions, like that of Washington, Oregon, and California, are actually more sort of on the chopping block in time than even coral reefs. And some of the people that are reorganizing our thoughts about this really feel like it's the upwelling oceanography by really important cultural resources, so really important fisheries, the oyster hatcheries on the west coast, all the larval action biology that goes on at river mouths and things, and so that coastal environments are kind of considered to be somewhat threatened. So certainly this group has an enormous amount to contribute to the study of that.
How do you deal with the synergistic effects of low oxygen?